Phospholipid bilayers inside of the cell, or surrounding the cell rather, act as selectively permeable barriers to separate things inside of your body. That's what membranes do. They isolate regions from one another. And the cell membrane is no different. It just separates the intracellular side, the stuff inside of the cell, from the extracellular side, the stuff outside of the cell. So, because of that, there has to be a way for things to move from the outside of the cell to the inside of the cell, or from the inside of the cell to the outside of the cell. So there has to be a way to transport things across the cell membrane. Now, when we talk about membrane transport, you often think that things can just move right across the cell membrane. So if we have this cell membrane, and let's say we take something and we put it outside of here, it's easy to think, well, that thing can just move from one side to the other of that membrane. But in reality, this very rarely happens because of that hydrophobic region inside of that cell membrane, right? So inside of here, remember that this is a hydrophobic region. So because of that, very few things can actually pass through that cell membrane unassisted. And those things tend to be very small particles or oil-based particles. So some drugs can pass through the membranes readily, and some insecticides even can pass through them. Right? But very few things inside of our body can just pass through the membrane. Glucose, for an example, can't just move through the membrane. Sodium, as an ion, can't just move through the membrane. They need help in order to do so. So they use what we call membrane transporters that move things across the membrane. And there are different types of transporters that we can talk about. So I'm just going to draw a very simplistic cell membrane here. And the first type of channel that we'll talk about will look something like this. And this is known as a gated channel. Gated channel. And what makes it gated is this little portion down here, this little ball on this little thing will swing, and it will basically close that channel off. Now, what can happen is that this protein, remember this is going to be a protein, can undergo a conformational change, and that ball sort of swing out of the way and open that channel up so that something, whatever it may be, can move through that channel. Okay? Over here, where it's gated, it can't move through the channel. It's blocked. Okay? So this is what we refer to as a single gated channel. It has a gate on one side, and in single gated channels, the gate can either be on the intracellular side, as I've drawn it here, right? If we consider this to be intracellular, this to be extracellular, or the gate can be on the extracellular side. It can be either. Gated channels can be even more complex in that they can have two gates associated with them. This is a double gated channel. It has two gates. And so I've drawn one on the extracellular side and one on the intracellular side. And in order for things to move through this channel, both gates need to be open simultaneously. Here, this gate is closed. Okay. Other channels can just be open channels. Let me draw this in gray here. So draw my membrane here. And let's just draw this one in purple. And these channels look like this. So whatever is wanting to move through it can just easily move through it. Okay, but one of the things that we have to remember is that channels themselves are selectively permeable. So what moves through here, we might say green can move through there, but this big orange particle gets blocked. Okay, it cannot move through the channel, or the charge inside of the channel might not interact well with the channel. Okay, so whatever is going to move sodium, that channel that's responsible for moving sodium, can't move chloride, can't move potassium, right? There is a level of specificity, a high level of specificity associated with these channels. Okay, so we have our gated channels and we have our open channels. Now, with these channels, some of them can be membrane transporters that act as carriers. And so carriers work a little bit differently in that, we'll draw this one in purple, okay, that it will sort of, let's draw another membrane so I got a little bit more room to work with here. These carrier proteins will sort of look like this. Look a little bit like this. And so what happens with these carrier channels, carrier proteins, is that a molecule will come in and it will interact with this carrier. And then what happens is that it will sort of reverse in nature, like so. Like this. Really close here, but open down here. Oops. Like this. So what happens is that when it reverses sort of its orientation, is that the red particles then can move in this way. So that's what a carrier protein looks like. Okay, so you have your single gated channels, your double gated channels, your open channels, and then you have your carrier proteins. Now, when we look at this, these proteins can move things in a variety of different ways, right? And so we tend to talk about their movement as being either uniport, which moves one thing, one direction. Okay, so it can only move things in a single direction, and it can only move one thing, right? We talk about symport channels, and symport channels can move more than one thing, but they move them in one direction. Okay, so they move things in the same direction. Your symport channels move more than one thing, but they move in the same direction. So this is like a sodium glucose channel. It moves sodium and glucose, but it moves them in the same direction. Your channels or your proteins can also be antiport channels. Antiport channels move more than one thing, but they move them in opposite directions. So can you think of an example of an antiport channel that you've talked about before previously? The sodium-potassium pump, right? Or the sodium-potassium ATPase. Moves sodium in one direction and moves potassium in the other direction, right? So that is an example of your antiport channel, okay? So these are the different ways that things can move in and out of a cell or across a membrane, right? And so in the next video, we'll talk about the mechanisms that the cell or the body uses to move things along with these channels or through these channels. So stay tuned for more.